I study ocean conservation, policy, and how everyone's knowledge can contribute to a better understanding of our environment. As a graduate student, I get really specialized training in how to do a very specific type of research. But the Engage program has given me another type of training that graduate students rarely get. And that's how to talk about my science in a way that's engaging, enlightening, and informative. You can learn more about our innovative program at engage-science.com. I also want to mention that the best way to keep up with Town Hall's programs is to become a member. Membership offers downfront seating, discounts on book and ticket purchases, and a mailed copy of Town Hall's monthly calendar. And it provides the support essential to bringing more than 400 events annually at affordable ticket prices. And now to our second guest, Sarah Pickett, a neuroscience PhD student, will share her research on hair cells, the specialized sensory neuron used for hearing. She'll explore how these tiny cells, their function, death, and regeneration, and the surprising ways zebrafish have informed her research. Pickett will also discuss how zebrafish research will help guide developing therapeutic strategies for restoring human hearing. Please give a warm welcome to Sarah Pickett.
All right. Thank you very much, Jillian, for the warm introduction, and to all of you for coming to my talk tonight. So I'd like to start by asking the audience, by a show of hands, how many people here know someone who has hearing loss or are experiencing hearing loss themselves? So I don't know if you all want to look around the room, but that's a lot of us. So that was a lot of you that raised your hands, and this fits really well with what we know about the prevalence of hearing loss. So in the US, hearing loss affects about 1 in 5 people. The Hearing Health Foundation estimates that hearing loss is the second most prevalent health issue in the world. And this is really amazing because our therapeutic strategies for curing or restoring hearing loss are really limited. So before we dive into research on hearing and the causes of hearing loss, I'd like to start by telling you a little bit about how hearing works normally. And to do that, I'll take you on a brief tour of the ear. I realize that this is a little bit anatomy heavy, so bear with me. I promise it won't last too long. Okay. So, we'll start with the outer ear. This is the fleshy part of your ear that directs sound waves into the ear canal, shown here. The outer ear ends at the middle ear, which is made up of 3 bones, and these bones vibrate in response to sound waves entering the ear. The middle ear bones vibrate against the inner ear, another bony structure, causing fluid movement within that structure. And in particular, the vibration causes fluid movement in this spiral structure, which is called the cochlea. And the cochlea is the part of the inner ear that's devoted to hearing. If we could zoom in on the cochlea and peek inside, we would see the actual sound sensors of the ear, which are called hair cells, here depicted in green. Hair cell is kind of a confusing name. They're not at all like the hairs on your head, but are rather very specialized neurons that respond to sound. The hair cells have a very particular shape. They have sort of hair-like projections on the top that we call a hair cell bundle. And on the bottom, they have a structure that allows them to communicate with neurons in the ear, here shown in red and yellow. These neurons send information that they gather from the hair cells to the brain. The hair cells line that spiraled cochlea in rows of 4, which you can more easily see if we look at the top down at the hair cells. So here, the hair cell shown in the schematic would correspond to this row here, and the 3 hair cells shown here would correspond to these 3 rows here. Again, we're looking at the top down, so we can see those hair cell projections or bundles very nicely. And to give you some perspective, each V here is an individual hair cell. So I'm kind of harping on the hair cell bundle, and the reason I'm doing so is because it's really important for how hair cells function. So it turns out that fluid movement in the cochlea actually leads to a mechanical stimulus or a physical deflection of the bundle that the hair cells turn into an electrical signal that they can communicate to their partner neuron. So in this way, hair cells are sort of like windmills. They're converting mechanical energy into electrical energy. Hair cells are super important for hearing function. So what happens when hair cells are lost? Well, it turns out that hair cell damage is a leading cause of hearing loss. So here again, I'm showing you a normal cochlea. You can see those nice 4 rows of hair cell bundles. And when the hair cells are damaged in the cochlea, it's not simply the case that the hair cells are kind of sickly looking, they're actually absent. So you can see in this region of damaged cochlea, there are really no hair cells. So you can imagine now how hair cell damage would be really devastating to hearing function. There are a number of different causes of hair cell damage leading to hearing impairment. This includes exposure to loud noise. It occurs as a normal consequence of aging. It can be related to certain genetic disorders and can be a result of certain medication side effects, which I'll talk about a little bit later in the talk. Hearing is particularly problematic for humans, and I want to illustrate that by using an audiogram. An audiogram is a graph that shows the loudness of sound on one side, here from soft to loud, and the pitch of sound on the other side, here from low to high. There are sort of everyday sounds plotted on this audiogram. So for example, the hum of a refrigerator is a relatively soft, low-pitched sound, whereas the roar of a motorcycle is a more high-pitched and very loud sound. You might have also noticed this gray banana shape on the audiogram, and this is called a speech banana. It's called a speech banana because it encompasses the sounds that we generate when we talk. So for example, the t of a T or the k of a K. If I now show you the pattern of hearing loss that's stereotypical, you can see that with just mild or moderate hearing loss, we very quickly lose the ability to hear the sounds that are encompassed in the speech banana. So this really impacts our ability to communicate. And if, like me, you have a grandmother that lives far away and I can only chat with her on the phone, you know that these kind of conversations can be really challenging. And this, for me, is a big motivating factor for understanding why hearing loss occurs and what we can do about it. To date, we have no way to really protect hair cells from damage or to regenerate hair cells that have been lost. So in my lab, we are using zebrafish to try to address these problems. You might not know zebrafish by name, but you might recognize them by sight. These guys are very common in personal aquariums. We don't house them in an aquarium per se, but you can see some tanks in our fish facility right here. That's me taking care of the fish, or beginning to. The zebrafish can lay lots of eggs. So these are newborn embryos that will develop into larvae. And the larvae develop quite quickly. So these animals are probably about 5 days old, and we can conduct experiments with them. So we don't have to wait very long once the fish are born to look at them. Zebrafish, in fact, do have hair cells, but the ones that we study make up a completely different sensory system, and this is called the lateral line system. The lateral line is used by the fish to sense fluid movement or water movement moving past the fish. You can see the organs of the lateral line in this image of a 5-day-old zebrafish with the head on the left and the tail on the right. This big black blob is its eye. And each green dot here is an organ of the lateral line that contains hair cells. So what do they use this system for? The lateral line is very important for fish behavior, like schooling, such as these fish in the image here, and for things like catching prey or avoiding predators, such as avoiding these sharks in this image. When the lateral line was first discovered, it was described as touch at a distance. So fish can use this system to sense their environment without actually touching anything. Okay, so back to the organs of the lateral line. If we could zoom in on one of these organs, we would see a cluster of hair cells. And if I turn this cluster on its side, you can more easily see that hair cell bundle, or those hair-like projections at the top. You might remember that these are quite similar to the auditory hair cells that also have hair-like projections at the top, or that bundle. And like the auditory hair cells, physical deflection of the lateral line hair cell bundle is converted into an electrical signal that they communicate to lateral line neurons. In this case, the information isn't auditory, but rather about fluid movement. So this brings me to a big question. Why study zebrafish hair cells? There are animals like mice that are really common in labs, and they have a cochlea, which would be more similar to humans, so why do we bother with the zebrafish? Well, there are a couple of really good reasons. One is accessibility. You may have noticed from the image that the organs of the lateral line, and thus the hair cells, are on the surface of the fish. This is in stark contrast to the cochlea, in which the hair cells are encased in that bony structure and then also deep within the skull, so they're much harder to get to. Because the hair cells are accessible, I can study them on an intact fish, so I don't have to do any dissections, and the hair cells are in their normal environment. Zebrafish hair cells are very similar to mammalian hair cells. They look very similar and they work in a similar way. And importantly, they are susceptible to similar kinds of damage. So I mentioned before, just kidding. Before we can talk about the different kinds of damage, I will first say that in order to protect hair cells from damage, we have to understand why damage occurs. The kind of damage that we're interested in addressing in my lab is the result of medication side effects. And the medication that we study is a class of antibiotics called aminoglycosides. So the name is not so important, but I will use it to describe the antibiotics throughout my talk. Members of this class of antibiotics have been used to treat things like tuberculosis, and they're more commonly used for pretty complex infections like sepsis, respiratory infections, or urinary tract infections. Obviously, antibiotics are super important for curing infection. We don't want to stop taking them. But it's unfortunate that they come with a side effect of hearing loss. And so we really want to understand why that occurs and how we might be able to stop that. So in order to study how aminoglycoside antibiotics affect hair cells, we can first choose an antibiotic. We can apply the antibiotic directly to the water that the fish are freely swimming in. And then we can examine the hair cells. I mentioned to you that we can study the intact organism. So in many of my experiments, the fish are alive. They are anesthetized. And we place them in a small chamber like this, and we stabilize them under a small net so they don't move around. So this is a chamber on a microscope. If I zoom out, you can more clearly see the microscope setup. And because the fish are alive, we can take pictures of the hair cells at different time points and look at how they're changing over time after the application of a drug. To give you an idea of the kind of data that we can collect, I have a movie that I'd like to share with you all. This is a movie that shows the antibiotics accumulating in hair cells after treatment. We can't normally see antibiotics, they're completely clear. But in this case, we've fused them to a colored molecule. So wherever you see green in the image, you know that there's antibiotic. When I start the movie, you'll first notice that antibiotic is accumulating in those hair-like projections, or the bundle, and then it will accumulate in the body of the cell. So I'll start this movie now. So you can just start to see the bundle and accumulating in the body of the cell, and accumulates so much that the hair cells actually explode. So I'll play that again. So in the body, and watch this guy, the hair cells actually explode. So aminoglycosides kill hair cells in the lateral line similar to the way they kill hair cells in mammals. And we can take movies of this, or we can look at snapshots of the hair cells. So here is a cluster of hair cells that's intact. It hasn't been treated with antibiotics. And here is a cluster of hair cells that has been treated with antibiotic. And you can see that all of the hair cells have exploded and disappeared, and there's only one hair cell left. And this is pretty dramatic because our treatments are only about 30 minutes long, so this happens really quickly. We can graphically represent the relationship between hair cell death and antibiotic treatment by plotting the number of hair cells that remain in a cluster against the concentration of antibiotics used, in this case from low to high. In a normal cluster, there are about 10 to 15 hair cells, so when there's no antibiotic, you can see that normal number. But as the concentration of antibiotics increases, the number of hair cells remaining decreases. This is a really consistent relationship. We can conduct this experiment multiple times and see the same result. And so we call this a dose-response curve. The dose-response curve is really powerful and cool because it allows us to test how the hair cells respond when we add different types of compounds that we will think will affect how the hair cells respond to antibiotics. So for example, if we add a compound that we think will protect hair cells from damage, it might have a dose-response curve that looks like this, where at each point along the plot, there are more hair cells remaining in the cluster than there were with antibiotics alone. We can also test compounds that we think will make hair cells more susceptible. And a dose-response curve for a drug like that would look something like this, where at each point, there are fewer hair cells remaining in the cluster. We can use the dose-response curve to test biological aspects of hair cells that occur when they're treated with antibiotics and lead to cell death. And we can also screen through hundreds of drugs to look for compounds that we think will protect the hair cells. And this is something that we've done in the lab, and actually we've identified a compound that does protect hair cells. So here again is a dose response curve. You can see the number of hair cells remaining on this side and the concentration of antibiotics on this side. And the solid line is the hair cells remaining after antibiotic treatment. In our lab, we identified a compound called Proto-1 that in fact protects hair cells when it's administered. So you can see in the dotted line the hair cell numbers remaining when Proto is administered in the presence of the antibiotic. So even at the highest concentration of antibiotic, only about 10 to 20% of the hair cells are lost. So this is really great. We hope that a compound like Proto or a similar could be used in combination with antibiotics to allow patients to take antibiotics and fight infection without causing deafness. Okay, so Proto is super great if you have all of your hair cells, but what if some of your hair cells have already been lost? Can we replace hair cells that aren't there anymore? Well, zebrafish put us in a really interesting position to study hair cell replacement because of the way that they respond to hair cell damage. So here again is a cluster of hair cells shown in red, and after we treat with antibiotics, you can see there's only one hair cell left. But if we wait just 3 days, all of the hair cells regenerate. And this is really remarkable. The fish are really good at it. And to illustrate that, I have another plot to show you. So in this case, we're looking at the hair cells remaining, but in this case, we're looking at regeneration cycles on the other side of the plot. A regeneration cycle is 3 days long, so this includes treating the hair cells so that they die and waiting for them to grow back. If you look at the plot, you can see these points. The hair cells are intact. We treated with antibiotics and they died and they grew back. We treated with antibiotics and they died and they grew back. And you can do this over and over and over. A member of my lab did this 10 times, so that was 30 days of treating fish and waiting for their hair cells to grow back, and they were able to do it each time. So where do these new hair cells come from? The new hair cells are generated by these green cells surrounding the hair cells in this picture called support cells. And we think that support cells function sort of like a stem cell. We think that when hair cells are damaged, they send a signal to the surrounding support cells telling them, hey, we're sick, we're dying, we need you to make new hair cells. And this would allow regeneration to occur. We really want to identify what the signal is because we think we could use it in the auditory system, coaxing cochlear support cells to make new hair cells when the hair cells are lost. So today I've told you a little bit about hair cell death in the zebrafish and a little bit about regeneration. And we hope that by studying fish, we can develop therapeutic tools to help protect and regenerate hair cells. So with that, I would like to thank the graduate program in neuroscience, my lab for sharing some of the data I shared with you today, NSF for funding my graduate research, the Engage program for a wonderful quarter and a great class, to Town Hall for the opportunity to speak today, and to all of you for coming. Thank you very much.
Yes, I was wondering if you could describe what Proto is.
So that's a good question. I don't know exactly the structure of the compound, but it is a drug that you would theoretically or ideally be able to take maybe orally in combination with an antibiotic. Can you elaborate a little bit on your question? Were you?
Yeah, I wondered if it was something that you've already constructed and that you're applying.
Oh, yeah. So I was not or specifically involved in this project, but members of my lab worked with some chemists at UW, and they modified different chemicals in different ways and small variations and tested each one actually by screening them on zebrafish. And they are working on similar compounds with a company to see if to use in trials to see if it's something that would be feasible to use for humans. Yeah, thank you.
Could this regeneration technique work with other causes of hair cell loss?
Yeah, so that's a great question. Ideally, it would work however the hair cells died. They're not there, so coaxing the support cells to become new hair cells would ideally work across the board. Maybe the strategy would change a little bit depending on the cause of death, but ideally it would be.
the chart that you had with the speech banana and hearing loss.
Yeah.
And it showed that we lose hearing kind of in those lower frequencies, right? Or is that just because there are fewer hair cells dedicated to something like that, or is it just that?
Yes, so it actually the pattern of hearing loss actually corresponds well with the way that the hair cells are lost. So I didn't really mention this in my presentation, but the hair cells are tuned to different pitches. And so the pattern by which your hearing is lost is related to the pattern by which the actual hair cells are lost.
Do you have a sense of how close you are to identifying that compound within the stem cell that would regenerate?
Oh, to a signal to help regenerate the hair cells? Unfortunately, I would have to say probably pretty far. We don't actually know what that signal is yet. But we have some ideas about how it could be communicated. One possibility is that it's something that the hair cells release and travels to a nearby support cell. Something that may not have been clear in the cartoon is that the support cells actually touch the hair cells. So it could be the case that as soon as a hair cell disappears, the support cells know that it's missing. And that's just something fundamentally different about zebrafish than humans.
Could you touch on how the antibiotic exposure low to high compares to what you see in people who are taking antibiotics? And did you measure just exposure or actual content of antibiotics in the fish?
So as far as how it relates to people, I'm not sure. I'm sure that people have measured levels of antibiotics that circulate throughout the body. I'm not sure that anyone has ever measured or has been able to measure how much antibiotic gets into the ear. I can say that the antibiotics are more likely to cause hearing loss, I think, when they're systemically applied. So you'd be taking it, let me try this again. You'd be taking it a lot or for a very serious infection, so it's kind of a last resort. As far as this works for the fish, again, I'm not sure that we've calculated the actual amount of antibiotic in the cell, but we do know that it enters the cell, and we know the amount of antibiotic that we're adding to the water. So the higher concentrations are, of course, more detrimental. I'm not exactly sure how well that translates to circulating levels of antibiotics, though. Okay, thank you very much.